Okay, sort of quick run through the anatomy, general anatomy of the brain. So we're going to have, I usually suggest to divide the whole brain story into kind of several parts. I give students this. So you see this pink stuff. The pink stuff. This is cerebrum. We're going to put this on the board, don't worry, okay? Don't write it. So this is cerebrum. This part right here, we're going to go through each part separately. This part right here is Diencephalon. This brown thing is cerebellum, literally small brain. Functionally, it relates more to the cerebrum than to anything else. And this is brain stem. Basically, as you go from cerebrum to diencephalon to brain stem, you kind of suddenly losing any conscious control over it. There's a lot of stuff going on, but you either aren't aware of that or you can't do anything about it. Does that make sense? Like, you may know what's going on, but it's out of your reach to modulate those responses. Clear? Some sort of shoe leather anatomy of the brain. So first of all, you see this folds and crevices, right? So, ridges and crevices. So ridges called gyri or gyrus, plural, singular. Okay? Yeah, it's Greek. So if you're curious if gyrus is cognate with a foot, gyro, yes it is. Saying, yeah, it's like roundish, wrap. And then sulci or sulcus are the crevices, okay? They really don't represent any deep functional importance. Are you following me? But there are certain brain functions that can be attributed to a particular gyrus. Like cingulate gyrus is the part of the memory formation. Like, we know that certain, so we can just say it, okay? So far we good? This is just, 2 name parts.
Can you repeat what those are again?
Can't hear you.
Can you repeat those again, what they are?
So, gyri are these ridges, elevated parts on the surface. Sulci are depressions like this crevices. Okay? Good? Clear? 2 hemispheres, right. I'm showing you the front of the brain, obviously. Right hemisphere and left hemisphere. Okay? Separated by the longitudinal median fissure, also known after one of my students named it on anatomy exam, the butt crack of the brain. The hemispheres are separated from cerebellum by transverse fissure. Fissure is the common term. It means really deep crevice. Okay? Each hemisphere consists of 5 lobes. I'm going to use just one hemisphere. Frontal, occipital, parietal, and temporal. I can ask you, I will ask you this on the exam. Now, I can reassure you 100%. It's not a tricky question. So I'm not going to, for instance, I'm not going to put the arrow here and say, which lobe? Right? Like if I want you to answer frontal, it will be like this. If I want you to answer parietal, it will be like this. Okay, good. What about the fifth one? The fifth lobe, which is insula, are located deep to four others. It's like within, so we can't see it here on this model. That makes sense? It's kind of a basic anatomy. We can put it away. And finally, start talking about, I'm going to kill this stuff, because I probably will need the entire space here. We're going to talk about cerebrum. Okay? So this pink part of the model, as a matter of fact, consists of three main substructures. Perhaps the biggest substructure is cortex. Cortex is the outermost layer of the brain. Okay? Some folks refer to it as gray matter. Second structure is white matter. And the third will be basal nuclei. Now, you're going to learn what these are a little bit later. For now, I want to do, bless you, a quick comparison between the cortex and white matter, and then focus your attention on cortices. I'm going to give you a hint. Well, remember neurons, right? Remember different parts like dendrites, soma, axon, right? What is the function of an axon? What does it do in neuron?
Takes it to the next one.
Transmits the signal. Very good. And we mentioned that it's where electrical signal is going, and in order not to lose that signal, in order to make transmission fast, what do we need to do? We need to put in insulation. Yes. We need to insulate it with a myelin sheath. Yes. Myelin is white. So what do you think white matter consists of then? Axons. Axons. So if axons transmit the signal, what is the function of a white matter? Transmit the signal, yes. Communicate. Now, since only, I'm kind of stretching it really thin, but since axons are white, bodies of neurons and dendrites are gray, and the function of a soma, remember dendrites receive the signal, right? Dendrites receive the signal. And then all those multiple signals, what happens to them on the soma? Yesterday's class was trying to find, and they understood what they wanted to say, but they couldn't find that fancy word. They get integrated. All signals, they get processed and integrated. Does that make sense? So this is why, so far, you kind of have to think about it this way. If you see gray matter, that's the thing that gets all the signals, processes them, formulates the response, okay? And then this response is being spitted out to white matter. White matter delivers it wherever. See what I'm saying? So cortex of cerebrum is, in essence, your consciousness on the large scale. Okay, does that make sense? Like, this is your conscious life. This is why you writing whatever I say, memorizing this, and you'll do it in a different way. We'll expand on that. So far, any questions? Basal nuclei, we'll talk about them. Extremely interesting structure. So far, any questions? Okay. There are 3 types of cortices, and we're going to start with the motor ones. So, quite obviously, motor cortices control your movements. Okay? So now we encounter again that example. Remember we were talking about in the very first, almost the first thing that I drew for you when we started to talk about nervous system, that it's unidirectional. Like CNS sends the information to the muscles. It never gets, like, motor response cannot go back from the muscles. So motor cortices send the signals to the muscles. We clear? 4 cortices here. First one is primary motor.
Never was primary.
Primary, yes. Primary motor. So what does it do? It directly controls muscles. Okay, so this is the team captain, okay? This is the team captain that tells everybody else, we're going to do some stuff. I don't know how to explain it better. I'm not allowed to use military terms because I never served. Okay, so that's directly. These neurons of primary motor cortex, through some relay mechanisms, they control muscles. Now, before we move any further, I'll have a question actually. A very small question. If you forget about all ethical problems, what would be the easiest way to learn what different parts of the brain do? Forget about humane treatments, forget about ethical problems. Which 2 very common tools that you can find in every household you need to study what different parts of the human brain do? If you go, you can get them at Home Depot, Lowe's. You need a box of nails and a hammer. That's all. This is called a loss of function. This is how brain was studied until the invention of MRI. Not that people were just, putting the nails in somebody's brains, obviously, but you have a patient with a brain damage. And you look at which part of the brain is damaged and what this patient can't do. Sometimes the other way around, you find an interesting neurological deficit and then you try to figure out which part of the brain is screwed up. Now we don't do that. Well, we kind of do, or we kind of know. What we do is called a functional MRI. So you can put a person in an MRI machine and measure the blood flow to the brain in the real time. Okay? So if you're memorizing, you're going to have increased blood flow to hippocampus and amygdala. If you are reading, there's going to be some frontal eye field and probably Wernicke's area. So, you can see the changing patterns of blood flow. So far, so good? So we're going to constantly mention what happens if this doesn't work. So, what happens if primary motor cortex is damaged? Let's say as a result of a stroke. What you cannot do. Very simple. Move. You can't move. You're paralyzed. And as a matter of fact, motor cortices, all of them are located in the frontal lobe. You have to know that. So one of the most common types of stroke is the occlusion of so-called middle cerebral artery. Middle cerebral artery supplies motor cortices. This is why so many people who experience stroke are paralyzed. Does that make sense? Now, second one is so-called premotor cortex. Okay? So this cortex does not control your muscles directly, but let's put it in the way of simple word skills. It's not a perfect description, but basically your premotor cortex formulates the movement. Does that make sense? When you are writing, you produce a very specific moving pattern. So that moving pattern is formulated by your premotor cortex, which sends it to your primary motor cortex, which then sends those signals to the muscles. Does that make sense? So if you will imagine a hypothetical case with the isolated damage to premotor cortex, you can move your fingers, but you can't write. Does that make sense? Or you can move your fingers, but now you cannot play piano. You used to be able to play piano, now you can't. Very important difference. If primary motor cortex is gone, you're not going to be moving. That's all. If this cortex, like not the whole thing, but some parts of this is gone, you can relearn. That's really important. People can relearn those motor skills. Good? Next one, I don't think requires any explanation. It's called frontal eye field. What does it move? Your eyes. Consciously moving your eyes. It's really interesting to see how evolution affected. It's a separate part of the brain, substantial part, that is so important because our eyes are so important for our interaction with the world, right? And it actually, in some people who experience stroke, their eyes often stop moving. They can't move them. And that results in an interesting condition known as functional blindness. Imagine that one eye in a patient works. It can move, it can perceive. Another one just stares straight or wherever. So pictures don't match, don't overlap properly. Does that make sense? So brain will shut down receiving the information from the damaged eye. It's still normal in a way. Does that make sense? But brain says, to hell with that. I don't need that information. It screws everything up. Okay? And finally, Broca's area. You control speech muscles. I want to make it very clear. We're not talking about comprehension of speech. Or knowing a specific language. Does that make sense? So for instance, patient whose Broca's area is damaged, they can write to you a perfectly readable and perfectly, a good text, but they have trouble saying it because they can't control their speech muscles. That part, does that make sense to everybody? Okay. Moving on to sensory cortices. There's going to be 6 of them. 3 high level, 3 low level. So first high level is somatosensory cortex. And there are 2 somatosensory cortices, primary and association. Now let's pause for a second and let's understand, because we're going to see that distribution. So like primary somatosensory cortex and association somatosensory cortex. What's the difference? Well, first of all, let's figure out what kind of sensation is that. Soma, what does that mean, the word? Body, very good. So, stuff that comes from your body. Things like touch, pain, temperature, vibration. For instance, I don't see my fingers, but for now I know that they're completely extended, right? You can see them. How do I know? Because I have receptors in my joints and muscles. I don't have to see my leg to know that I'm standing on one and another one is bent in the knee, and I can even know to what degree it is bent. See what I'm saying? So this is all somatic sensations. So what does primary somatosensory cortex do? It receives kind of the basic qualities of that information. Probably the easiest way will be touch. Lukewarm, not really rough, but not absolutely smooth, okay? And shape is kind of circular, okay? So those are kind of qualities I can even appreciate to some extent the size, okay? I know that it's my hand that's touching, not like, and it's my palm, not the dorsum of the hand, you see? So all of that primary. What does association cortex do? It puts all the qualities kind of together and allows me to understand what I am holding. Okay, so you probably all have the keychains and you have multiple keys on them. And you probably, if you reach for them, you can tell apart your car key, okay, your car remote maybe, if they separate, maybe even keys from your house or apartment and keys from some work stuff and all that kind of things, right? You don't have to look at it because you know how they feel. See what I'm saying? So this ability to basically create an image, recognize an object using touch is the function of association cortex. People with the damage to this, they can touch it all right, but they will have to look at it to understand, okay, now this is my car key, okay?
So would they get determine like round or square, like they wouldn't be able to feel that? Like they can feel something
They can feel, it's smooth,
Okay.
it's warm, it's my palm. I have no idea what the hell is that, okay? And here's the thing, look, I used that example yesterday. If I hypothetically imagine I have a bucket, like that bucket in front of me, and I stick my hand and I have no idea what's inside. And I touch it, and it's slimy, kind of flat-ish, with a really something kind of, feel like it has a body and 4 legs, and it's slimy, and it's kind of flat, and it has a soft belly. It's probably a frog. See what I'm saying? But if I've never touched a frog, and I have no idea what frog feels like, I won't be able to do that. I'll just say there's something live, and then slime and all that kind of stuff. So you do have to have some patterns to compare with. Does that make sense? It's going to get more and more clear, like visual cortex. By the way, somatosensory, it's a parietal lobe. Visual cortex is occipital. And it also has primary and association. So primary, it will tell you the contrast, the colors, the brightness, shape to some extent, movement. But then, here's the thing, look, it is black, it is large, and it is moving towards me, and it is moving very fast. All of this I can apply to a German Shepherd, right? Or, I don't know, Ridgeback, or any other dog breed. But then association cortex will put all the specifics together and will say, yeah, I know that that's my German Shepherd. And she's really happy to see me. Or that's a Ridgeback that I've never seen, so I probably should just get going, as fast as I can. See what I'm saying? So that's recognition. The most striking example, really, is auditory cortex, which is in temporal lobe, also having primary and association. Now you will be surprised by the number of qualities that you have with your sound. What can you tell about a sound? You can tell 3 things. It's pitch, volume, and location, and that's it. You don't need anything else. Because then you combine it, basically you combine pitch and volume, and then you start recognizing what is this. Does that make sense? It's all. So, pitch, volume, location, that's your primary. What is this? That's your association cortex. Makes sense, right? Now, what are 3? And look, these are very high-level cortices, meaning that there's a lot of information processing going on here. Does that make sense? Which kind of can be explained, by human evolution, really, because we use these cortices like so much. That's our main way to understand the world, as a matter of fact. You see where I'm going with this? This is how we see a lot. 70% is visual. Auditory is super important because we talk, okay? We constantly touch things. Now, olfactory. Smell. It's all over the place. What do I mean by that? There are pieces of olfactory cortex in your frontal lobe, hippocampus, hypothalamus, amygdala, mammillary body. They are parts of Diencephalon, the olfactory cortex. So first of all, there is no primary association. It's kind of a more holistic approach, But think of it this way. We suck at smells, at olfaction, compared to many other animals. But we still, you'd be surprised, we're going to talk about it, we can distinguish a lot of smells. And smell is one of the most powerful memory producers. Okay? I can give you an illustration from our family, my personal experience with the family. The first state that we came was Nebraska. Okay, don't ask what, different story. But we came to Nebraska and we step out of the plane, it has very specific smell. It was great, it was good, but just very specific, very different from my home country. Okay? So we live in Nebraska, we moved to Louisiana. We step out of the car in Shreveport and totally different smell, completely different. Okay, fine, we live there 8 years. And then all cases are in the summer when smells are in full bloom. My wife and I would fly here to Cleveland to find a place to live, like a few days. And in the summer, I step out of the airport, tell her, this is Nebraska. That's the Midwestern smell. Seriously, Ohio and Nebraska smell the same. This is Midwestern smell. South, Louisiana, Alabama, Mississippi, Georgia, different. My kids go to school in Tennessee. That smells more like Louisiana. Okay? And memories, it immediately triggers the memories. Like it was 9 years from the time we came to States. And immediately I remember us like getting out. First time in the States. So it's really powerful memory trigger. Two of the most boring cortices, gustatory and visceral. So those sensory cortices are in insula, so taste, and importantly, conscious appreciation of what's going on with your organs. I mean, you can, like, your urinary bladder is stretched. Your stomach, you have like bloating in your stomach is stretched. You have some understanding of those feelings, right? Does that make sense? Location-wise, function-wise, you know. So, again, primary sensing integration, primary sensing integration, primary sensing integration. Good? Okay, the third category of cortices are association. There are three of them that we're going to talk about. So, I'm going to call it prefrontal. It's also known as frontal association area. I mean, obviously it's located in the frontal lobe, just the name says, okay? So frontal lobe. And this is you. Okay? Like your demeanor, the way you study, like you like to study 2 hours a day, but for a longer stretch of time, or you cram everything in 3 days before the exam. That's you. No, seriously, okay? You timid, really outgoing, you a visual learner, you need to touch it with your hands, that's all there. This is your personality. And we know that for multiple loss of function situations, the most famous is the British guy who got a rod through his frontal lobe and survived, and they pulled it out and he changed completely. But in my opinion, more interesting sort of story is lobotomy. You've heard about that procedure probably. So it was used to mostly treat patients with schizophrenia, and in my humble opinion, it actually worked. Because people were cured from schizophrenia. They were not schizophrenics anymore. They were not themselves, though, also, in an absolutely pure term. So the person that would sort of come out of lobotomy procedure is not the person who went in. You see it?
Procedure.
Huh? Lobotomy. Removal of certain parts of the frontal lobe.
Oh.
Basically removing the parts of prefrontal cortex. So this is why it was unethical. You know, like you put Johnny in, whoever comes out is not Johnny, bless you. It's a totally different person. You see what I'm saying? So, this is how we know. Wernicke's area, parietal lobe. Speech recognition. Your ability to understand what I'm saying is there. As my ability to say whatever I'm saying. Okay? That makes sense? And this is why you probably know about, you know, bilingual children who kind of have an advantage. Brain is kind of, it's not a muscle, but you get the drift. The more you exercise it, the more it develops. Basically, on the structural level, neurons build more connections between themselves. And it's known that children that grow in multilingual environment, their brain works better. That's it. Make sense? Believe me, adults who start learning language, a language, they benefit from this. That's great. Okay? And finally, limbic system. I'm going to save myself from talking about limbic system later, so we'll touch on this now. Conscious recognition of emotions. So, you're angry. You're angry at your boss, but you don't want to show it because you understand that if you do, your boss can fire you. Make sense? Or you got a new position, you come for the last time to your work, and you just flip your boss off because you don't care anymore, right? You're crying or not crying. I'm not talking about physiological responses. You see what I'm saying? Like, physiology is separate. We'll get there, okay? Limbic system does it also. So where is it located? It's everywhere. So all over. And for instance, an important part of your limbic system, as we will learn, we'll go back to this idea, your brainstem and your hypothalamus. Okay, because this is unconscious stuff. So for instance, your probably the easiest. You really, really like a person. Just really like, not as a friend, okay? So when you encounter that person, it's your decision whether to show your affection or to hide it. That's the conscious part here. Does that make sense? But when you see this person, you can't control your increased heart rate. You can't control the dilation of the pupils. Okay? Or you see someone despicable, your pupils will constrict. No, seriously. Okay, so, does that make sense? So it includes hypothalamus, it includes amygdala, all those, a lot of various structures that produce that emotional response. Okay? Questions? I told you we're going to talk about some fun stuff. I mean, diseases. So, Alzheimer's. I have good news for everybody. You're all going to have it. The main question is what's going to happen first. Alzheimer's or death? Alzheimer's is the disease of old age. Go ahead.
But is it like hereditary? Because there's a lot of elderly people in our family.
I'll touch on that. Hold on a second. It's a really good question that you asked. So what is the mechanism? First of all, what is the mechanism? So in Alzheimer's disease, your brain, two structures are formed. One are called beta amyloid plaques. And another one is tau protein neurofibrils. So these structures, plaques and neurofibrils, are not supposed to be there. Plaques are like blobs. Neurofibrils are really, really long, tangled fibers. Are you with me? They mess up the structure of your brain. They mess up the connections between the neurons. Does that make sense? So usually it manifests mostly as the dementia. Okay? We'll get to possible origins. Okay? Anatomical, on the gross anatomical level, the size of the cortex shrinks, and the size of the ventricles, usually there is a mild ventriculomegaly. They a little increase, okay? What happens there? Short answer, I have no idea. There are some genetic risk factors, okay? But one of, since my main passion, big passion, is microbiology. Of course, I'm like those people who try to talk about their favorite thing whenever. There's several studies now demonstrating the pretty strong link between various infectious agents and Alzheimer's, like herpes or bacteria that cause gingivitis. And one hypothesis that I really, by the way, another reason to brush your teeth. One hypothesis that I really like, and it makes a lot of sense, is that this is normal. This is the way for your brain to contain pathogens. Does that make sense? Your brain, when pathogens accidentally get into the brain, and in this case, you're right, it's really hard to get there. But if they do, your brain tries to stop them, creating those structures, trapping them. But it is a collateral damage. Make sense? When it accumulates, well, it's probably there's some threshold, and then that's it. Obviously, it's not the only cause for any means. Okay? There's no cure. There are treatments.
There are treatments.
Very unsuccessful, like really. Very poor treatments. One interesting observation that was made is that the amount of nicotine equivalent to 3 cigarettes per day reduces the risk of Alzheimer's by about 25%. I wouldn't be so optimistic. There's some other stuff. Okay, it doesn't have to be. They don't have to be cigarettes. Any nicotine supplying stuff works. But honestly, it was shown multiple times. The best way to reduce the onset, to delay the onset of Alzheimer's, is put your brain to work. Any form. You don't have to do theoretical physics at 70 years of age. Sudoku's fine, crossword puzzles, memorizing rhymes, learning language, anything is fine. Just put it to work. That makes sense? Now, this is bad stuff, and this is pretty common stuff. Another disease that isn't so common, very rare, terrifying, is prion disease. And it's Creutzfeldt-Jakob's disease, CJD. Okay. So what are prions? Prions are proteins that are found in your brain, and they're normal. There are some bad prions, unfortunately. Are you following me? And this is typical Walking Dead, zombies, whatever you want to call it. When a zombie bites a normal person, what you get in the end? You have 2 zombies, right? When a bad prion interacts with a good prion, you have 2 bad prions. Make sense? The more bad prions you have, your brain turns into a sponge, almost literally. The fancy term, spongiform encephalopathy. So pathology of the brain, where your brain looks like a sponge. Original discovery was in Papua New Guinea, where they looked at, there was a local tribe with a phenomenal, I think we all should practice that custom. They showed respect to the deceased elderly by eating their brains. They thought that they will acquire the knowledge. All they acquired were prions, and they years later developed the same condition, died, and their brains got eaten, and the tribe was on the brink of extinction. And when scientists figured it out, they said, dude, you got to stop that. They did, and they survived, really. Then there was mad cow disease in Britain, and some unfortunate folks ate, long story, but basically from meat. In the United States, there is wasting disease of deer, mostly in Wisconsin and Minnesota, and I don't know about now, there was like sort of a spike in this, and game warden had to inspect the game to determine whether or not it looks like it's like emaciated and stuff. But basically, don't eat nervous system. Don't eat brains, don't eat spinal cord. That's the main part. There is a genetic disorder that's called familial hereditary insomnia, but I may be wrong with one word there. You stop sleeping, and 2 months later you die. That's all. Yeah, pretty bad. Yeah, but the main problem with prion diseases, there is no cure, and unfortunately there is no treatment. It's palliative care, so you can't delay. It's just, they can, go ahead.
Do you think the brain is really this size or is it smaller? Because when I saw autopsy be done, I thought that the brain, it looked a little smaller. I don't know if it's because he was dead.
Not sure. I can give you an average volume.
Okay,
It's 1,400 cubic centimeters, a little more than a liter,
okay.
like a quart and a half, okay? It's probably a little bit bigger. This is most likely a tad bigger than that, okay? We can try to put it in the Benjamin one day and see if it fits. Because Benjamin is kind of life-size-ish, okay? Maybe a bit bigger, not really. That makes sense? So this disease now can be diagnosed. Before it was just post-mortem. But I don't know what's the benefit of it. Like knowing what you're dying from? No. Questions? Okay.